Well, to start with, I wish everybody here present this afternoon a very welcome to this official ceremony. And therefore, I open officially this ceremony, this academic ceremony, in which Mr. Yuandi Zhao will defend his academic thesis entitled Multimodal Analytical Strategies in Drug Discovery. May I invite you, Mr. Candidate, to present a summary of your studies and the conclusion of your thesis in the next 50 minutes. And I give you the word.
Okay. Thank you, dear Proctor, committee members, supervisors, colleagues, friends, and family. Thank you all for joining me today, either in person or virtually, for my defense. In the coming 15 minutes, I will present my thesis entitled Multimodal Analytical Strategies in Drug Discovery. To start with, I would like to briefly introduce the pharmaceutical R&D pipeline, which can be summarized into 5 stages, including discovery and development, preclinical and clinical research, CMC development, registration, and post-market safety monitoring. This thesis is mainly focused on the first stage. In this process, we always face a lot of challenges, such as long time consuming, the difficulty of molecular modification, unwanted effects, and the application of technologies. Generally, to discover new drugs, we focus on certain diseases, which are usually caused by their disease-related targets, in most cases, the proteins. And we want to find some lead compound can inhibit these targets. So the first protein target I researched on was 15-lipoxygenase 1, or 15-LOX1. It can catalyze arachidonic acid and linoleic acid and lead to different diseases, such as inflammation, vascular disease, neurological disease, or obesity. So therefore, we want to find some compound can block this pathway and hopefully can remediate this disease. So based on the published 15-LOX1 inhibitor, we picked some promising scaffold and side chains, and then synthesized 67 compounds as potential 15-LOX1 inhibitors for further research. So with this newly synthesized compound, we first need to screen them to check whether they can inhibit 15-LOX1. Here we used the E. coli bacteria to express 15-LOX1 and used it to screen these compounds. In the end, we found 20 compounds showed inhibition tolerant to 15-LOX1. For the next, we need to do the assessment of this compound. As we prefer to take medicine for the disease instead of poison, here we first test the toxicity of this compound with MTT method using cellular model. The MTT chemical can interact with only live cell and form purple crystal, which means the more purple the color is, the more cells are alive, and the less toxicity of the compounds. However, the cells are usually too simple to represent our complicated body entity. Therefore, we also used C. elegans to test the toxicity of these drug candidates. With this model, we can evaluate the health of both parent and progeny, and also the broad cells. It can give us better understanding of the toxicity studies. Besides the assessment of toxicity, the assessments on the observation, distribution, metabolism, excretion are also important. And one key factor can influence this process is the lipophilicity. If the lipophilicity is too high, the drug molecule will easily get into the cell, causing unwanted cellular target. Or the drug molecule will be too difficult to be pumped out of the cells, leading to the risk of toxicity. Or the molecule might be uptaken by the cells and affecting the pharmacokinetics. To study these molecules, liquid chromatograph or ILC and mass spectrometry or MS can be used. In an ILC system, the molecule can be separated in a reverse phase ILC column according to their lipophilicity. Molecule with lower lipophilicity will be first eluted, and therefore we can get the relationship between molecule lipophilicity or log P and their retention behaviors or log K, and thus we can get the molecule property information with ILC. As for the MS, we are going to talk more about the electrospray or ESI in this thesis. The injected molecule will be either positively or negatively charged in an ionization source and then enter mass analyzer. such, for instance, the quadrupole, where only certain ions can pass through it as setting, collision cells, where the ions can be fragmented, IMS and TOF, where the ions can be separated. The first two together are usually called tandem mass spectrometry, or MS-MS. In the end, the molecule will reach the detector, which gives the mass-to-charge ratio, or MORZ value, as molecular structure information. With these technologies, we found that the log P of our newly synthesized compound were too high, which unfortunately makes them difficult to be druggable. Therefore, we need to do the optimization of this lead compound. An effective way to do so is using cytochrome P450 enzyme, which can modify the molecule by adding OH group on it. With this hydroxylation, the molecular hydrophilicity and polarity will increase, and the lipophilicity will decrease. As we demonstrated before, the hydroxylated molecule has lower lipophilicity and will elute earlier. Therefore, they can be separated in the assay, and MS can detect the MORZ value changes in the mass spectra. In the end, we found that among 5,000 CYP450 mutants, 200 mutants show the hydroxylation bioactivity, and therefore they were used to catalyze the newly synthesized compound as mentioned before. And hopefully, this metabolite can be used as a next generation inhibitor of 15-LOX1. To study these metabolites, besides LC-MS, we also used the MS-MS to evaluate the selectivity of these mutants. With this technology, the lead compound can be first analyzed to show where the scaffold and sidechain fragments, and the metabolites can be analyzed by MS-MS to show where the OH group were added. As we want the mutant can function on this compound with known position, so we can optimize this lead compounds with demanding goals. Therefore, with the MS-MS result, we can know about the bioactivity of these mutants, and hopefully they can be used as a biosynthesis tool in drug discovery. So far we talked about the molecule and we talked about protein target, and we know they can interact with each other. Therefore, it leads to the next question: how can we evaluate about the kinetics of this interaction? A gold standard for this is to use surface plasma resonance, or SPR. In this system, these targets are usually immobilized on an SPR sensor, and the drug molecule will be injected. Once the interaction happens, the surface plasma will change, leading to the shift of the peak minimum angle. And this shift can be monitored by an SPR detector and given as sensorgram. So in the sensorgram, we can clearly track where the association and dissociation phases and use them to calculate the kinetics. So we know that SPR gave us the kinetics of molecular interaction in real time, and MS gave us structural information and can realize both quantity and quality. In this research, we are thinking a way to combine them together as new drug discovery platform, especially for drug screening. However, the buffer used in SPR usually has high salts, which is not suitable for MS. So based on our previous study, we found that 4-isomorphyllene acetate, or 4-EMA buffer, is suitable for MS measurement and leads to lower charge states. Moreover, it doesn't influence the bioactivity of the enzymes. So for the next step, we want to know whether this buffer will influence the kinetics, and it might be a solution to couple SPR and MS. To address this question, we used a target named indolamide 2,3 dioxygenase 1, or IDO1. It can lead to immunoescape, and several inhibitors have been reported can block this pathway. Therefore, we also used them for this study. So we first compared the kinetics of IDO1 and its inhibitor using 4-EMA and PBS buffer. We found that the kinetics are comparable between these two buffers. And with this result, we developed the SPR-MS method in both offline and online approach. In the offline approach, we first measured the inhibitor directly, and then measured the sample after flowing through the SPR. And as for the online approach, we couple the outline of SPR to the inlet of MS directly. The offline results show that During the association phase, most of the inhibitors were bound to either one. And during the dissociation phase, the bound inhibitor left, which we can see from the mass spectrum. We could see the increase of the peak intensity. Also, we used a non-either-one inhibitor, diclofenac, and the opposite result was observed. So for the online results, we just saw the signal in both SPR and MS so far, and which just prove the concept and need to be further development. So we know that SPR can perfectly used for the study of molecular interaction, but how about the cellular interaction? As it gave us direct insight into the cellular response to the drug molecule and gave us real-time pharmacokinetics information. Therefore, to develop the SPR cellular assay, we captured the cells on an SPR sensor and used it for the SPR measurements. In this study, we focus on the GPCR, which is on the cell membrane and can interact with chemokine, for instance, the interleukin-8 or CXL8 or IL-8. And this interaction can lead to the different biological processes. And invention 3, which is a small protein from tick, has been reported can be binding to CXL8 as potential inhibitors. Here, we first used the SPR study to study the kinetics between CXL8 and invention 3, and used MS to confirm the complex formed in their mixture. So with this, we used the SPR sensor cultured with endothelial cells and inject the CXL8, invention 3, and their mixture to observe the cellular responses to these different injections. As shown in the results, we found that the endothelial cells showed the response to CXL8, but not to the invention 3. As for the mixture, the signal of the mixture were lower than that of CXL8, but higher than that of the invention 3. This result demonstrated that the invention 3 can inhibit the interaction between CXL8 and endothelial cells by forming the complex with CXL8. And this also indicated that the possible application of SPR in cellular interaction study. So as a conclusion, my PhD research showed the drug discovery process focusing on different drug targets, including 15-LOX1, EDO1, and GPCR, and showed the way to discover and evaluate the lead compound as potential inhibitors. Also, we gave the idea of lead-to-lead optimization using CYP450 mutants. We found one interesting compound can be used as potential 15-LOX1 inhibitor. And we showed the using of multiple technologies such as assay, MS, and SPR for the molecular study. And also we showed the method development, including the integration of LC-MS and SPR-MS, which can give us multiple information with one run. And as for the SPR cellular assay, which could also apply as a new strategy in drug discovery. So with all of that, I would like to thank to my supervisor team, my students who contribute to my research, the collaboration among different universities and companies, fundings, and thank you all for your attention. And I would like to give the word back to the prorector. Thank you.
Thank you very much, Mr. Candidate, Mr. Zhao, for your clear and very detailed presentation, I must say that. Before we start, I'm going to introduce to the audience 5 members who are present here. First of all, I introduce myself. I'm Professor Steinbusch, the pro-rector, and I'm assisted today by Professor Oru, who joins me here. But more importantly, we have the promotion team who are sitting opposite of me. And from the left side, from my side here to the right side, I would like to introduce Professor Honing, who's here. He's Professor in Substance Imaging at our university, first promoter. Professor Dijkgraaf, who is in the middle. She's Professor in Biomimetic Chemistry. And also Dr. Hadavi, who is affiliated with the Department of Analytical Sciences. And they have worked together with the candidate for a very long time, almost 4 years, to bring them here that he can defend this afternoon his thesis, and that's why we also like to recognize them. For the audience, today there will be 5 opponents. 4 will be here present on site, and one opponent is online, as you would see on the screen, but later on the big screen where he will appear as well. So without further ado, exactly at 50 minutes past 1 o'clock, we're going to start the discussions, and we're going to start with the chairman of the assessment committee, Professor Heren, and I would like to he's professor in MS imaging at our university, and I'd like to give him the opportunity to start the discussion.
Thank you, Mr. Prorector. Mr. Candidate, before I start our discussion, I would like to compliment you with a wonderful piece of work, culminated of research in this booklet. And I particularly want to compliment you not only with the beautiful artwork, but also quoting one of my favorite physicists, Stephen Hawking, on the back. Of course, in my congratulations to you on the completion of your work, I would also like to congratulate your promotion team who has guided you along the way. But now let's get into the discussion, which is why we're here for today. And your thesis deals with a number of multimodal molecular imaging strategies. And as your primary promoter often emphasizes, definitions are important to get clear. So I would like to discuss with you some aspects of your definitions in Chapter 1 related to your structural analysis tools. Firstly, I would like to ask you if you are familiar with UPAC and the 2013 recommendations of definitions of terms related to mass spectrometry.
Highly esteemed opponent, thank you for your kind words and the questions. Unfortunately, I'm not familiar with that, but I would like to study more about that.
I actually prepared two further line of questions. One way you said, no, I don't know, and the other one, yes, I do know. But no, I don't know. So in that case, can you describe me what you think are, in your mind, the most important terms when describing tandem mass spectrometry or MS-MS?
Okay. So I think the MS-MS can give the fragments of the molecule, so we can somehow analyze the structure as the MS give us the structural information. So that's why I think the MS-MS is really useful for that structure analyze.
Okay, I'm going to stop you, interrupt you here for a moment, because you're now explaining to me what MS-MS is capable of doing. That was not my question. My question was, what are the most important terms to describe tandem mass spectrometry? And you already named one, which was fragments. Can you elaborate a little bit on the terms that describe tandem mass spectrometry? If one of them is fragment ions. You mentioned fragments already.
Yes. And also I think, based on my knowledge, I think it can be also used to know about molecular interaction and how strong they bind to each other. So I think that also can be a term.
So dissociation energy could be one. And the selection of an ion to start with, how would you define that?
Sorry?
The selection of the ion you're going to fragment, how would you call that ion?
It's called a precursor ion.
A precursor ion, great. Now we're there where I want you to be. Unfortunately, in your chapter 1, on page 23, you have figure 5, and all of a sudden I see you're talking about daughter ions. And you talk about fragment ions, and you talk about product ions. What's the correct terminology? I mean, you use all these terms intermixed, and I'm confused. What is the right terminology to use?
So actually, I think, based on my knowledge, I think the daughter ion fragments or the product ions, they are referred to the same things. So it's based on the precursor ions. After fragment, it will lead to that. But also, I read like the paper, like if you call it for the daughter ions, maybe you will call the precursor ions parent ions. So that's the answer for this. So
exactly. So the proper definition, according to UPAC, are precursor ions and product ions. And as I said, you use them sort of intermixed in your thesis, which, you can use different terminologies to describe the same thing, but good definitions are important, right? So, and UPAC sort of defines those. Let's continue a little bit on the sequence of events in tandem mass spectrometry, because you already talked about that a little bit. And you mentioned selection of ions and fragmentation and observing the product ions. In that chapter 1, you described this hyphenated strategy that you have abbreviated LC, UV, Vis, MS, 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 IMS to further resolve structural fragments.
Yes.
Can you briefly sketch to me what here is new in this strategy in light of how you have defined MS-MS?
So I think the new of this is like we integrated all of them together. So as I demonstrated before, with one injection, we can possibly get multiple information of that, which I think maybe in the academic world is not very new, but for the industry it's really new. And I think there are more more application of this technology can be transferred to the industry as well. So such as
I'm going to interrupt you here because otherwise I run out of time for my other questions. But I would argue that in your abbreviation, you use one MS too many. And I don't think it's an all-in-one strategy because you either do LC, UV, Vis, MS, and that's it. And then you do another experiment, the way I understood, but correct me if I'm wrong, your thesis. And then you do LC, UV, Vis, MS, MS, and then IMS, eye mobility spectrometry to characterize the fragments. Do I interpret that correct or am I wrong?
Actually, no, because like for the UV and MS, we use a splitter so we can get the UV information and the mass information directly. And also if we setting it correctly, we can set the MS and also get the IMS information altogether. So it can be separated to...
It can be, I understand, but have you done it?
Yes,
Have you done acquiring all the same sample at the same time in one experiment?
Yes, I did up to the MS. So for the IMS, I just collect them separately, but still it can be setting as altogether.
That's also how I interpret it. It can be done, but maybe sometimes you do things separately. Let's move on. And I was told by the pro-rector to prioritize my questions, so I will move on to chapter 6, where I got really excited because you were using native mass spectrometry in combination with SPR. And in figure 2 on page 90, you show a native mass spec spectrum of the CXCL8 evescent 3 complex that you also showed in your presentation, and its constituents. Now, if I read the text belonging to that figure, apparently you were a little disappointed about the abundance of the complex. But you do not explain what you expected. Perhaps you can describe to me now what level of complexation you expected in that spectrum.
Yes, so for this one, because we want to say how the evescent 3 will inhibit to the CXCL8, so we hypothesize that they will form the complex. So that's why we use MS to test whether the complex was formed. And from the mass spectrum, we can see the complex formed in there, so which is also why we use the mixture injected later for the SPR cellular assay.
Understood. But you state, and I'll read this out loud, despite the relatively low abundance of the complex compared to the response observed for CXL8, the presence of this complex is evident. Great. But you talk about the relatively low abundance, so you expected higher abundance? So what did you expect?
Yeah, because originally we expected that the complex will have high signal, but I think the reason we got this signal is because of the in-source fragmentation of the MS system. So that's why we can say, or maybe the interaction of the CXL8 and evescent 3 are not complicated. So that's why we can still see the CXL8 and evescent 3. And also,
that's exactly. Sorry to interrupt you, but I want to make this a little bit more efficient. That's actually what I wanted to question you about, because you blame dissociation, but you don't talk about association. And I was wondering if you could use that native mass spectrometry experiment to determine the KD instead of determining the KD with SPR as you've done. Could you elaborate very briefly on how you would do that with native mass spectrometry?
So I read about this use MS to analyze the kinetics, but the problem is like we need to prepare all of the samples because it's time dependent, and that's why the SPR can give us real-time measurements. So I think there is still difficult and challenges for use MS to track the kinetics. But yeah, there are more space to discover, I believe.
Okay.
But
actually, I'm slightly in disagreement with you because there are many publications that demonstrate the kinetics of protein-protein complexation using native mass spectrometry, and that's what I was hoping where I would take you. But I think my time is up looking at the pro-rector, so I want to thank you for this lively discussion and give the word back to the pro-rector.
Thank you.
Thank you very much, Professor Hera, and indeed you were sharp on the 9 minutes. My compliments. We move forward and we go to Professor Holman, and he is going to start with you online. May I have Professor Holman on the big screen? Thank you very much. Professor Holman is professor in Biocatalysis at the Technical University in Delft, and I would like to welcome him and thank him that he joins this afternoon. And I'd like to give him the word. Professor Holman.
Yeah, thank you very much, Mr. Pro-Rector. Dear Mr. Candidate, no, nomenclature is an issue. Thank you very much for this very interesting thesis. I learned quite a lot. You know, it's a firework of analytical methods, so it was really interesting for me to see. But you can imagine that my questions will be a bit more on the biocatalysis enzymology side of things. And I would like to start with chapter 2, where you actually show an AlphaFold model of the lipoxygenase. So that emphasizes a bit the importance of, basically artificial intelligence in silico design on drug design and on enzyme design. So I was a bit astonished that you used the structure, but more or less only as a decorative element. I did not see too much of docking, of investigation on how these molecules could interact with the lipoxygenase. Could you elaborate on this a little bit more?
Highly esteemed opponent, thank you for your kind word and question. So yeah, indeed, we didn't do much about the downstream regulation pathway research of this, because also like we try to do the docking of this compound with the enzyme, but however, like the docking result is not very good, and also so far there's no crystal structure of this 15-LOX enzyme. So we mostly use the rabbit one, which have like 87% homogeneous of this one. So that's also might be the reason why the docking result is not very good. And also that's also our next step, because we want to find some lead compound for the 15-LOX one, and we need to do the optimization of this. So I think with the next generation of the inhibitor, we will do the downstream regulation research as well, such as use the cells or animal model with the transfer of the LOX15 genes, yeah.
So taking the risk that the discussion might be going into a completely different direction, I just looked at your thesis and the word docking appears once in your thesis. So apparently, and that's also what I remember, you did not report your docking results. You mentioned that the docking results were not very good. But what do you mean with not very good? Did they not show you the results that you were expecting, or was the quality of the docking so bad?
Because I'm not a specialist of the docking or the software engineer, and this is the collaborate with other faculties. So the docking result actually showed, based on what I understand, the score is not very good. So which means like it can show the interaction between this molecule and the target, but not ideally. So that's why we said, okay, this might be not very good for this compound. But also the reason why I say this is a promising compound, because we used the in vitro enzyme screening and we found the lower the IC50 values, so which means they can interact with each other. That's the reason for this. Yes.
That's an interesting point. So you've been focusing on the development of suitable inhibitors, of course. I fully understand. But your docking was not successful in that way. And don't you think that this is also interesting information that also should or could have been included in your thesis? Or do you think that a non-positive result, whatever positive means, should not be published?
So I still believe the result from my thesis, that's I can say for sure. But also, like, because as I mentioned, I'm not a specialist of the docking, so I'm not sure how they do the docking with them. And also I learned that the docking is usually really difficult to mimic the whole biological environment. So that might also lead to the fail of the results. But I can't say for sure about that. Yeah.
But my opinion would be it's also negative results are relevant results, and I find it a bit a pity that you have decided against making your results public. That may lead to someone else later on doing the same mistake, if you want to say so. So to my mind, it should also have been included in your thesis. But it's a very nice thesis, so I'm not really criticizing this at all. Maybe a more question on the understanding. And here you also double check for the cell toxicity using the MTT assay. And I could not find too much information about the modus operandi of the MTT assay in your thesis. Could you quickly elaborate why purple color is linked to viable cells?
So because the MTT is a chemical can only interact with live cells, so which means, like I demonstrated before, the more purple the color is, which means more cells are alive in the well, and which means with that.
What is different between live cells and dead cells that make the difference in this color?
Because the MTT can only interact with live cell, so it can't interact with the dead cell. So which means the more purple crystal formed, which means the more cells are alive, because the dead cell won't form the purple crystal. So that's we can use the MTT.
Do you have any idea about the molecular mechanism of this interaction? What happens with MTT in living cells?
So, yeah, this one I can't say for sure, sorry, because I think it's the chemical reaction with the cells, so it must interact with some live cell things. So I'm not sure about it, sorry.
No, thank you very much. I think I still have 1 or 2 minutes left. So then I would like to switch to chapter 3, where you identified a few mutants of a new P450 monooxygenase. And I was lacking any sequence information about these mutants. So you show in which plate these mutants were identified, but you don't show what the mutants actually are. Is there a reason behind it, or did you just not sequence them?
Well, this is a collaboration with a company, a pharmaceutical company, and also this sequences is confidential, so that's why we couldn't put here. But we just want to prove like they have this power and they have this biosynthesis tool for the drug discovery.
Meneer de Prorector, do I still have time for one question, or? I hope so.
Okay, no, there's no time. Thank you.
What a pity. Thank you very much. I give the word back to you, and I'm very satisfied with the answers.
Unfortunately, with my microphone, I cannot communicate directly with the onliners. That's the problem. Good. Then we continue to Dr. Kuykens. Dr. Kuykens is Scientific Director and Fellow at the Unit of Drug Metabolism and Kinetics at Janssen Pharmaceutical in Beersen in Belgium. And I would like to thank him that he joins us today live, all the way from Belgium, I would say, and I'd like to give him the word.
Thank you, Mr. Prorector. First of all, dear candidates, I want to congratulate you and also your promotion team with a very nice thesis. It's covering a very wide variety of topics, so it also illustrates that you had to learn and master a lot of different skills. That's also, for that I also want to congratulate you. First of all, I want to continue with the discussion of the previous jury member on chapter 3 on the mutants, cytochrome P450 mutants, where I was wondering, so you used this 5,000 and you end up with 200 finally mutants. I was wondering how the mutants differentiate themselves from commercial kits that are available from companies like HAFA and Codexis. And yeah, you have, let's say, a lot of kits available and mutants from yeast, from bacteria, etc. So how is this set of mutants you used unique or different from these, let's say, commercial sets?
Esteemed opponent, thank you for your kind word and questions. So if I understand correctly of your question, you ask about how can we say the mutants can be specifically used for the drug discovery.
No, I was more wondering, so it's not the only set of mutants on cytochrome P450 that are available. Yeah. There are already other mutants available, let's say sets available that you can buy. So is this set different from these, or how do they compare? Do you have different reactions, for instance, that you can do with these that are not readily available in the outside world?
Okay, so first, as I mentioned before, the mutants were designed by the company, so I don't know the details of this, unfortunately. But also, because the basically principle for these mutants is to add the OH group to do the hydrogenation of this compound. So that's generally basically principle. And with all the mutants, it have the same function. And we want to know, we also have the wild type to compare with. So we want to know whether this mutant can act better than the wild type, and also with known position. So we want to increase the effective of that mutants. Okay. And also because the CYP450 is original from our human body, so that's why we use the one from the bacteria, which we can produce more and use for the industry world. Yeah.
And so you selected 200 more active ones from the 5,000 you tested. So how do you see the next steps? So now you have 200 mutants. So if I want to synthesize like say an hydroxylated compound, do I start screening all these 200, or do I start with the most active one, or do I combine? So what's the next step?
So I think the idea is based on the customer request, because with as demonstrated in the chapter 3, we want to know where the OH group was added, either on the scaffold or the side chains. So I think based on the customer request or based on the drug discovery goals, they can use certain mutants for this design, because we already clarify how the mutants function on the molecules. So that's the next step if we based on the request of the design goals.
So does it mean, do I correctly understand you, that if I need, for instance, an aliphatic oxidation at a certain position, okay, I have to select this and this mutant to produce that? So yes, with a high success rate?
Yeah. So I think it's also in the table 1. So we showed some mutants can function on different positions. So that's why we categorize them.
But you tested that on a few compounds. It's not that you have a set of hundreds of compounds, let's say, where you can validate that a certain reaction only works with a certain mutant.
So also this, because based on the at the very beginning when we screen the 5,000 mutants, we select some variety compound, so such as steroids, also the long chain. Sorry, I'm not the chemistry, and also the with some aromatical rings, so we can use a really variety of the compound to detect them.
So, and if you want to make a certain amount of compounds and you have one mutant, how can you upscale? To which levels can you upscale this reaction? Can you go to the kilogram amounts, or it's just milligrams? Or
Oh, this is a tricky question. So I'm not sure because I like the knowledge from academic to transfer to the industry. So I'm not sure how much amount we can go, but yeah, okay. Sorry for this.
Yeah. So let's switch gears. In chapter 2, you describe the integrated analytical approach to identify compounds, and I was particularly interested in the CE50 that you described, so the survival yield at 50% survival yield for a compound after fragmentation, and use that for DHEA and testosterone. You illustrate that, but of course you have the standards in hand, then it's easy to do it and see which one is fragmenting more easily, but how do you in practice use that for a new compound where you have to identify a structure? How do you use this CE50 yield?
Sorry, I didn't quite get
So the CE50 yield, so it describes how easy a compound fragments and also gives information on structure, so I agree with that. But if you have an unknown and you would fragment it in a mass spectrometer, you can calculate the CE50. But how do you use that to give you structure information? Is it something you have a database with thousands of compounds and you do machine learning? Or is it something you say, okay, likely, I know this is the class of compounds and based on the CE50 you have to more or less think in this direction or that direction? Is it, how do you use it to define a structure from the CE50 in practice, let's say, if you don't have the standards in hand?
So I'm not very sure about this question, but based on my knowledge, I would say we can use this to check the... Sorry, I don't think I can answer this question, sorry.
So yeah, to give an example maybe, so you have the structure on page 21 of testosterone and DHEA. So if you would not know which spectrum belongs to which one, would you then, based on the fragmentation and the amount of fragmentation, can predict this is testosterone and this is DHEA?
Yeah.
And do you know why the DHEA is fragmenting more readily? It's more easily fragmented than testosterone? What explains, let's say, the higher yield for or lower yield for testosterone?
I think that it's based on the chemical structure, and because I think the testosterone has a conjunction structure, so that might be the more stable for this structure. But I think that can give us the information about that, like which molecule is more stable and the structure is more stable.
Do you know where you get where the most likely site of protonation is on these molecules? So the positive charge, where would the positive charge, where would it be positioned on both molecules?
I think it will be on the oxygen part.
Yes, yeah, they have two oxygens, but you probably mean the carbonyl oxygen.
Yeah.
Yeah. And so you have, on testosterone, you have a conjugated system, so it's likely stabilizing this positive charge, and that's why it's probably more stable.
Strong. Yeah, more stable, yeah.
And with that, I give the word back to the pro-rector.
Thank you. Thank you very much. Then we continue the opposition with Dr. Achten. He is affiliated with the Department of Biochemistry at our university, and I'd like to also give him the opportunity to discuss with you.
Thank you very much, Mr. Pro-Rector. Dear candidate, I would first like to congratulate you and your supervisory team with this nice piece of work. Broad range of topics, as already illustrated by the other speakers here, from analytical techniques to synthetic organic chemistry, protein expression, and cell culture. I'm a biochemist, and I would like to dive into the biochemistry of Chapter 6 with you. And there you attempt to develop a cell-based SPR assay. And as a model system, you choose the interaction between CXCL8 and evasion 3. And in my opinion, that is not the most easy system to start with, because you have a system with multiple receptors that are activated by multiple chemokines, and these chemokines are often present in dimeric or higher oligomeric states. Now, in your first experiment, you evaluate the interaction between evasion 3 and CXCL8. And on page 85, you even mention the interaction, binding affinity, and structural details of the interaction have been well characterized. Now you repeat this, and you find binding constants of 130 and 1.6 micromolar. These values differ greatly from the previous obtained experiments, which have binding constants in nanomolar range. Can you explain this difference?
Esteemed opponent, thank you for your kind word and questions. So actually this one we want to, because also I think the results I read from that is we used a different system as well. And also the kinetics will be influenced by the environment and also the setting and the temperature. So I think that's a reason why it caused the deliberation with these two different results. But also with this new SPR system, like why we want to use this is because it will be easier to operate, and we can also develop the new cellular assay. So yeah, it is. I do agree with that. The results are different, but with the instrument measured in our system, we say this is another kinetics. And also, as I demonstrate here, like the interaction is not only with CXCL8 and the evasion 3, it's also between CXCL8 and CXCL8. So they form the dimer. So that also might influence the kinetics here. So, yeah.
Well, I agree. I don't fully agree that the systems are different, because they're both SPR systems that these constants are measured on. But let's move on. So you already mentioned you have homodimerization of CXCL8 present. Now, this chemokine may also form heterodimers with CCl2, CCl8, and CXCL4.
Yeah.
Did you take this into account?
No, because we used actually the pure sample of CXCL8, so I don't think there will be possible interaction with CXCL4 or CXCL others.
I thought you were going to answer that, but I see two sources of where these chemokines might come from.
Yeah.
Can you now maybe think of a source where these chemokines may come from?
It's from the Biochemistry department, and it synthesize the CXCL to themselves. So it's actually from the old
But you
stocks.
culture endothelial cells on the chip, and these endothelial cells can themselves produce chemokines.
No, actually, I think the reason of that is like, I think it will express the CXCL8 based on some stimulation, if I remember correctly. So that's why, but under the flow pressure, we just used it for the CXCR there, the receptor there, yeah.
So if we then ignore the endothelial cells as a source, in your cell culture, you also add 10% FBS.
Yes.
Do you know what FBS is and what that contains?
Based on my understanding, I think it's supply the nutrition for the cell to grow.
Not only nutrition, but also chemokines and cytokines. So that might also be a source where you're influencing your results.
Yeah, but I think in the SPR measurement during the buffer, we didn't put the FBS in that. We put the HEPs there as a buffer. But during the culture, we do use the FBS for the cell growing.
Exactly. So if I look at your curves, there is a lot of standard deviation in the curves on page 91, figure 3, and 92, figure 4. Do you think this could be related to either heterodimerization or homodimerization of CXCL8 here?
I don't think so, because this curve is like typically for the SPR cellular assay, and as we immobilize the cell on the surface, and also the cells, they are alive, so they might need to shaking or wigging, so that's why it might cause the frustration of this curve. But based on the fitting curve, we can clearly see the trend of this, the interaction between them. So yes, that's.
So say I believe you now. So all of these factors that I previously mentioned have no role in this, and the only thing you're seeing here is binding to CXCR1 or CXCR2.
Yeah.
Did you actually check if these cells produce these receptors?
So yeah, that's a good point actually, because based on our previously planning, we were planning to test the expression of that. But also I found some paper like under the pressure of the fluid system, it will start to express a CXCR1 too. So it's actually on the surface. And also based on the different responses of the SPR cellular assay to CXCL8 or invention 3, we can see the difference. So definitely there should be the CXCR on the surface. And yeah, that's the answer for this question.
Do you know what the under what circumstances an endothelial cell will normally produce CXCR1 or 2?
Sorry?
Under what circumstances would a endothelial cell normally produce these receptors?
So it's under the fluid pressure, so that will start to express that. I read like after 5 minutes under certain pressure, it will start. And that's why we also used the BPR, back pressure regulator here to cause some like pressure in the system, so we can guarantee that the CXCR1 are expressed. But that's a good point. Actually, I think if we can double check the expression of them, that would be ideal for this research. I agree with that.
Do I have time for one last question? I think the receptors are mainly produced under inflammatory conditions because these cells want to attract neutrophils to. Did you also try to stimulate these cells with an inflammatory stimulus, or can you think of an inflammatory stimulus?
Yeah, so that's a good point. Also, it's like the coming story of these chapters actually, because we not only want to see the interaction between the protein and the cells, we also have the like I would say big hypothesis. We want to also see the cell-cell interaction. So that can be actually the next step. We want to use the neutrophilic cells to co-culture them all together to see the interaction among different cells even. So yeah, that's actually the next goals for this research.
Or as an intermediate step, just add TNF-alpha to give the cells an inflammatory boost, get the CXCR1 expression up,
yes,
and then see how your binding curves change.
Yeah, that's a great suggestion. Thank you very much.
Thank you very much. I give the word back to the co-director.
Thank you very much. Then we moved to the final opponent, which is Professor Oru. He is professor in organic chemistry, but also the director of the Aachen Maastricht Institute for Bio-Based Materials. And I'd like to give him the opportunity to discuss with you.
So thank you, Mr. Pro-Rector. And I would like to congratulate you, dear candidate, with this beautiful thesis. And I'm also really happy to see that there's also at least quite some organic chemistry involved. So that gives me also the chance to discuss with you perhaps some aspects of that. The first, let's say, thing that I would like to discuss with you a little bit more in detail is actually on page 27, where you have sort of summarized a schematic diagram of the, let's say, lipoxygenase process with these heme or this iron type enzymes that do a peroxidation or peroxygenation. So if you look at the process from arachidonic acid, starting from arachidonic acid to these products, then it introduces the peroxy radical at the C15 carbon atom in the chain. I know that if you do peroxygenation at the C12 position, then you it can be followed up in the body by a cyclization reaction, forming a 5-membered ring, which then is that, let's say, the precursor for making prostaglandins, which are hormones involved in inflammatory processes. So there was quite a big drama some 20 years ago of a big pharmaceutical company that introduced this type of, let's say, inhibitors of this cyclooxygenase as a treatment, as an inflammatory, let's say, treatment. But the drug was withdrawn because of heart failure of many patients that took it. So your system seems to be a solution for that. And you're also referring to some toxicity studies with C. elegans. So I was wondering, to what extent can these toxicity studies with this, let's say, C. elegans also point out the heart problems? Or would this potentially be also a risk for these type of, let's say, structures targeting on this LOX?
Highly esteemed opponent, thank you for your kind word and questions. So yeah, actually we use the C. elegans because we can also monitor the health of the parent and progeny and also the broad size. So we actually found something interesting with the result, even we didn't put it here. Like, for instance, we found like with the incubation of some compound, there are more eggs with not hatching. So which means it might be influence the speed of the development of the progeny. And also, because we also talked about the broad size and the progeny, we research on that. So we can also say with this result, we might think there might be some neurological toxicity of this compound to the progeny. And yeah, so I think the C. elegans gave us more information than the cells itself.
Okay, thank you. But it's probably still some studies still to follow up to, before you can decide whether this is really working or not, I guess. Yeah. So maybe to go more into some, let's say, how you designed your molecules that is based on this scaffold hopping approach. Yeah? And for that, in fact, you have described that in more detail on page 34 and also on page 36, and later on there is also a big table on page 39. So when I was going through this as an organic chemist, I see that you are, let's say, basically changing the core structure, so introducing an extra nitrogen or an extra oxygen in the core structure. So that's the scaffold that you change. But you then decorate at the outside of the molecule at the primary amine. So would it not have been easier if you look at your synthetic schemes, for instance, on page 36 or one of the others, to introduce more functionality in the aryl side chains, which also would be, let's say, making your molecule more hydrophilic, and then your synthetic routes would be a bit more short, but you would get similar information?
Thank you for the question, but unfortunately, I'm not the expert of chemistry, so this is also a collaboration among different universities. But based on what I understand, I think that's a good suggestion, actually. We can do that to have more hydrophilicity compound. But also this scaffold and sidechain were picked from the published 15-LOX1. So we compared the different library and say, oh, this might be promising for this inhibition research. So that's the reason we picked them up. But I think that's a good suggestion if we can choose something with more lower lipophilicity.
Okay, thank you for that you agree with my suggestion. So maybe one final question. I think there's still some time. And for that, I would like to ask one of the paranymphs to read out proposition 3 for me.
Biosynthetic hydroxylation using cytochrome P450 offers an environmentally friendly and regioselective alternative to traditional metal-catalyzed hydroxylation in drug optimization.
Thank you. So, very interesting proposition. And if I look at the chapter 3, where these hydroxylations on the steroid scaffold are, let's say, a topic, I was wondering, you are mentioning in your proposition environmentally friendly and regioselective alternative. So, if you would look like what is another main feature of enzymatic or biocatalytic synthetic processes, is regioselectivity the only thing that you should think about?
And also, I think the efficacy of that as well, because like if we use the enzyme, we are aiming to have the fast hydroxylation reaction there. So that's also, if we can speed up the drug discovery pipelines, also save the time and save the money. So that's why we want, besides this proposition, we also want to speed up and also have high effective of this.
But if you think, let's say, about the stereochemistry, is that not also a very important aspect?
Dear Mr. Candidate, you are allowed to finish the answer. You don't have to. You also can say, I'm closing my opposition here.
I can quickly finish the answer. So I agree with testosterone. It's also fine. That's why when we selected the original compound screen is mutants, we set different variety of this compound. So it's also covered that category. Thank you.
Thank you very much. Dear Mr. Zhao, dear Mr. Candidate, the official time for defending your thesis has now passed. The degree committee will now withdraw to discuss not so much the quality of your thesis that has been approved, but we're going to discuss your presentation and the way you have defended your thesis this afternoon. And I request that you and your company await the results of all our deliberations and the return to the aula. Thank you very much.
The PhD defense has now ended. The degree committee will debate the candidate's performance behind closed doors. This process usually takes about 10 minutes.
Long road to the south side. 10 miles in my rearview mirror, I know what it felt like. My goal's only getting clearer, east side to the west side. No place like home, if it's questions that you've got, go the extra mile and die. The line, I know you know that I can't read your mind. Years can't fly by now, don't waste all your time. Cause I'll go the extra mile. Long road to the south side, 10 miles in my rearview mirror, I know what it felt like. My goal's only getting clearer, east side to the west side. No place like home, if it's questions that you've got, go the extra mile and die. On the dash, got my laces tied. Long road, I don't waste no time. Break rules because fate decides. With the team and we chase the light. I make a move, fall down, shake it off. I hate to lose, bad branch, break it off. No room for negativity, praise and love. Prepare for deep part cause we're taking off. Get the mileage, traveling so far from home. Gathering knowledge, other connections. I put the time in to get everything I ever hoped for. It's a challenge and we all in. It's a better future that we go for. And even though these nights seem long, I know that I am never on my own. 10 miles in my rearview mirror, I know what it felt like. My goal's only getting clearer, east side to the west side. No place like home, if it's questions that you've got, go the extra mile and that. Long road to the south side, 10 miles in my rearview mirror, I know what it felt like. My goal's only getting clearer, east side to the west side. No place like home, if it's questions that you've got, go the extra mile and that. Years can't fly by now, don't waste all your time. 'Cause I'll go the extra mile.
Dear Mr. Zhao, the degree committee here present has assessed not so much the quality of your thesis, but the way you defended your thesis this afternoon, and particularly also the presentation you have given this afternoon. In view of its positive verdict and taking into account your previous qualifications, the degree committee has decided to grant you officially the degree of doctor. Professor Honing is authorized to confer upon you this academic distinction according to Dutch university customs, and therefore I invite now your supervisor to take the floor.
Do you promise to work in accordance to the principles of scientific integrity at all times, to be careful and honest, transparent, independent and responsible?
Yes, I promise.
By the authority vested in us by law and in conformity with the decision of the committee here present, I hereby confer upon you, Yuandi Zhao, the degree of doctor and grant you all rights attached to custom and law. As evidence for this, I now present you with the degree certificate signed by the rector, the secretary, and the other members of the committee, and affixed with the official seal of the university.
I give officially the floor again to Professor Honing to speak out the official laudatio.
Dear Yuandi, dear Dr. Zhao, congratulations. One of your two dreams, and I will come back to number 2, has become true. Also congratulations on behalf of the other promoter, adding the modern medicinal chemistry to your research, and the co-promoter adding, I would say, sciences and being your teammate in all this beautiful work you did. But before continuing, a great thank you to the assessment committee here present, Dr. Stijn Achte, Dr. Philip Kuikens, Professor Ron Here, Professor Ouro, and Professor Frank Holman for reading and taking the effort to go through this thesis. And also many thanks to the collaborators in the matchup project, Pedro Hermkens, ex-Organon, and Peter Kwaadvlieg at Inusin, Samantha Hughes at the VU Floris Rutgers. And a special thank you to Elena Skruti and Professor Vitala in Finland helping us with the SPR, or helping you with the SPR and cellulary assay. Now that all nerves are gone, and just before a new episode starts in your life, here I will give you a laudatio, or better a sort of critical review of what happened in the last 5 years. Now a laudatio is about you, and it always starts, to my opinion, with reading your name, Yuandi. Yuandi can be referred to origin or place of origin, and it seems that there was a noble emperor of, actually the last emperor of the Han dynasty, who was called Yuandi, and he was a believer of Confucius. And actually that was also the reason of his death, because he was too kind. So nobility, honesty, and harmony were one of his virtues. Having said that, I was looking to the family name, and there is no real explanation of your family name. And only thing what I found is that your family name Zhao is linked to an emperor Mu Wang. And what the good thing was of Mu Wang is that he was going on journeys all his life. And of course he was killed because he was not present where he would have been present. Now, going into you as a person and not only your name, in search of really describing you as a person and scientist, and using only one word, I only know one word for you, and it's colorful. Colorful in terms of your great drawing and painting skills, or better artistic competences. And I remember Pailang, Daria and Helen trying to being impressed on your paintings and trying to persuade you to focus on an artistic career and not on scientists. Now, luckily for us all here, you continued, and the cover of your thesis, the presentation, and the defense of today are a perfect witness of your ability to combine both virtues in advancing drug discovery. Colorful as your hair was rarely the original black and could change to all types of colors, sometimes even combinations which I did not know were existing. Colorful as already in February 2020, only 5 months after being here, you painted yourself as a real Roger Rabbit or Bugs Bunny, fully involved in a Maastricht carnival with many of the other students. This is also a proof of your rather strong and large fantasy, your, let's say, little bit obsession and love for Lego and interest to construct imaginary worlds is an indicator for someone who can picture an ideal world. Both colorful and fantasy can be considered the foundation for enjoying life. I never in my life met a young PhD student who has been traveling around Europe more than all students of the UM together. The number of your Instagram reports, including all aspects of your journey, can be regarded as really extensive. Together with restaurant specialist Theo and others, you thoroughly tested nearly all restaurants here in Maastricht and even beyond. I tried but failed to keep up. Nice food and beautiful cities were combined with parachute jumping in the Czech Republic, relaxing on a high-speed boat somewhere between the Greek islands, and more. And the most famous are the Europe, the most famous trips you did is the Europe in 8 days round trip you did with Bailan. Yeah, so bus in and bus out, plane in and plane out. Of course, Amsterdam was one of the first cities to visit, while Barcelona seemed to be the preferred place in the last year of your PhD. I recall meeting you at the bus station in Maastricht, taking the same plane to Barcelona, and you planned to celebrate Christmas with Lydia and her family. My first thoughts were, poor guy, he needs to eat carbon, which is a tradition in Catalonia for people who did not behave. And I thought also, what will become of him? I witnessed in Instagram you smashing El Tío, and I will explain later for those who are interested, with a stick, really with all the energy you had. Not yet eating the carbon, so you kill the bees nearly and then you don't get the carbon. Upon your return to the Netherlands, I noted, without going into details, that in 1 week you learned all the bad words, both in Castellano and in Catalan. The role of Lydia here is not completely clear to the day of today, and luckily your continuous improving Dutch is much more refined in that sense. Twice I met you in the UM Sport Gym. You with a face of, what the hell are you doing here? And a little bit saying, scary, hello. The truth is, as I understood, your second dream was to have a six-pack. Yes. And after this encounter with you, I understood it was another six-pack, as I was thinking upon all the feedback I got from your colleagues. I will leave it like that. As a scientist, you broadened your scope and transformed from a cellular biologist and a molecular pharmacologist to, let's say, a drug discovery expert. Having knowledge on bioassays, a little bit of toxicology, of target activities, medicinal chemistry, lead candidate production, and especially SPR and SPR MS. You achieved a grant from the UM SWOL program and managed many, and you saw that, many students which were assigned to him. And when I assigned them to him, he had sometimes a very strange face, like not always a happy face. You came in the Netherlands with the intention to search for bioactive compounds in traditional Chinese herbs. We did a lot, but that's not what we did. You dared to step outside your comfort zone into the world of liquid chromatography, mass spectrometry, to understand the relation between molecular structures, physical chemical properties, and their behavior. You grew in confidence, and as a reply to your question 2 weeks ago to me, I hope I made you proud, my answer is very simple. Yes, more than proud. It was truly a pleasure to collaborate with you. Proud because just doing what I asked you to do in the beginning, You change into an independent scientist with his own thought and disagreeing with mine, Ingrid's, and especially also with Daria. But, and coming back to the question of the first opponent today, dear Dr. Zhao, although the FDA uses the word preclinical in pharma, it's, to my opinion, still lead optimization. So please believe me and take this as the truth from now on. Again, proud, as your natural way of engaging others was the basis to team up with Ingrid to make a next step in bioactivity and real-time pharmacodynamics, and that is the purpose of this SPR cellulary assays, and the collaboration with Daria and Professor Vitali on getting it to work. You discussed with BioNavis specialists on integrating a specific measurement cell in the SPR to measure it, and work together with Samantha Hughes at the VU to use the C. elegans, especially for reprotoxicity. Proud because you delivered work together with Kim, I hope she's here, which really could meet industrial R&D. You became a sparring partner for industrial hotshots, to my opinion, like Peter Kwaadvlieg, Monica Muller, Martin Schuurman, and Pedro Hermkens. Sometimes to be proud of, I would say. As mentioned, together with them, you proved the validity of the Aori cell lines, the lead-to-lead library approach, and the beautiful new compound which you showed, 16A. With support of Jos de Man of Crossfire, you researched IDO1, and I think IDO1 remains your beloved target. Your next bus station in life is Edinburgh, which to me, on first hand, did not make any sense. Even more rain, colder compared to the beautiful Maastricht. Yet, I think it's not relevant, as I assume your colorfulness and your fantasy will lead to more sunshine on those dark shores in Scotland. And even more likely, you will certainly spend at least 3 months per year outside Scotland in warm and exotic places all over the globe. To conclude, dear Yuandi, dear Dr. Zhao, I wish you a bright, colorful career and life in any corner of this planet. Be well.
Thank you very much.
Dear Dr. Zhao, does it feel good?
Helemaal goed.
Helemaal goed. Very good. That's excellent answer. I would also like to congratulate you on behalf of the board of directors of our university. And in my congratulation, I also take, of course, the promotion team who guided you. And I think they also deserve a part of the congratulations as well. I also would like to thank the members of the Corona, the members of your assessment committee who are here fully available and who prepared questions to you, maybe tough questions, some, and they were present here available. Some of them are online, and I also would like to thank him that he likes to join this afternoon. I also would like to congratulate maybe your family who is online available, who are watching now to the camera over there. You can see there, and who have now should be proud to the achievement you have made so far. And by saying this, I'm going to officially close the ceremony, in which I used my hammer, and I said we are closing the ceremony. How we are going to proceed? We understand we have a reception, and I think the audience already can go to the rafter. We will stay here and make a picture with the Professor Holman online. So I need 1 or 2 photographers to stay with us. Don't forget this. And then we make a picture on the stairs of our university before we also proceed to the rafter and join you over there in about 7 minutes. Thank you very much.
Thank you.
the stairs, we're not going to be that quiet.
Yes.
Okay, Professor.
If you want to address a few words or one sentence to Professor Holman, it would be nice. Maybe you can take the bill. So he has 3 hands, please.
Thank you for joining me today, and without your help, I couldn't make it today. So I'm really thank you for all the help and suggestion. Thank you.
Thank you.
Are you talking to me?